Hey, Bio30. So I'm going to do a video on topic 5, genetic engineering. And this is just a supplement to doing your notes in class. I'm going to go over the notes, and if you have any questions about the notes, then you can definitely send a remind or just ask in class. So to get started, what is genetic engineering? Well, anytime we change the DNA in a species, we are making a new DNA, which is called recombinant DNA, and that in terms is genetic engineering, and we call that recombinant DNA because it is a new DNA. How do we do this? Well, we need 4 things: a plasmid from a E. coli bacteria. A plasmid is just a circular piece of DNA from bacteria. We're going to need a gene that we want to put in, so required gene. We're going to need restriction enzymes, which will cut the required gene out and also cut the plasmid open to put the gene in, and then ligase, which will glue it all together. Why do we use E. coli? Well, bacteria like to replicate, and it's cheap to use, and so we can use it, and it's very small to use. So we can use it to create a whole host of things, which will be coming up in the notes. So here's a good graphic showing how we do that. You can see at number 1, we are cutting the plasmid, which I remember I told you was the circular DNA. And after we've cut it open, remember using restriction enzymes, we will take the required gene and insert that into the plasmid. Once we've inserted it, we'll use ligase to close it. And then once it's all closed, we'll put it back into the bacteria and allow it to replicate. But now you can see at the end it's replicating with the new gene and producing whatever that gene is programmed for. So an example here would be insulin. Okay. And there's number 3, using the ligase. And number 4, putting it back in. And then number 5, replicating. Now, why do we do this? Well, there's a number of reasons. The first one that we have here is for agriculture. Maybe we want to genetically modify some organisms, and in this case the example is tomatoes, and there's a flavor savor tomato picture down there, and it was supposed to resist cold weather and last longer, though it never went to market. Another one that is there is Roundup Ready canola. So this canola has been genetically modified to resist the herbicide Roundup, which Roundup, if you know, kills everything else except for the canola. So it'll kill all the weeds surrounding it, which increases crop yields and also reduces the use of other herbicides. Number 2 here, we could use it for human hormones and products. So we used the example of making insulin, which if you're diabetic, that's a definite must. Some other uses for human hormones are antibodies and proteins, interferons, and somatotropin, which is a human growth hormone. Number 3 here, the production of BST. So it's known as bovine somatotropin, and this increases milk production in cows. This is mostly in the United States, as it's illegal to use BST in Canada. Number 4, gene therapy. We can literally take out a defective gene and replace it with a non-defective gene. An example here would be cystic fibrosis or hemophilia. And we would just cut out the defective gene and put in the new gene, just like we showed you with insulin. So using the restriction enzymes and then the ligase, and hoping that it would get rid of the bad gene. What are some pros of recombinant DNA? Well, one example would be insulin. That's a huge plus. And maybe some production of disease-resistant strains, for plants or agriculture. Maybe you can combine the characteristics of two different organisms into one. So you can take the characteristics of one and put it into something else. An example that I've seen in the store lately is a grapple, where they have an apple and a grape together. Number four, this is quite common, selective breeding. If you have any type of pet at home, it was probably selectively bred. That's how you get toy breeds. So example, a toy poodle or a toy schnauzer. They do this in farming all the time for cows, to selectively breed the cows that produce the most milk. And then the last one, number five, cure human disorders. That's a gene therapy, so cutting out genes and putting new ones in. What are some cons? Some people think you shouldn't be playing with DNA, is the playing God or eugenics. Maybe now you're only selecting which genetic information you want to keep in offspring. So, could you theoretically just have certain traits that you want to keep in babies and have designer babies? Example, tall and smart and athletic. It's very expensive, and we don't know what the long-term effects are on other organisms, as genetic modification is fairly new. It's just started in the 1990s. How could we analyze DNA? PCR, polymerase chain reaction, which we don't need to know. But the two that I want to focus on is gel electrophoresis, which will separate the chunks of DNA into specific genes. And then we can do what's called DNA fingerprinting, which will match the bands of the DNA. And we're going to cut that DNA into several pieces, and then we can match it with a baseline. So we can skip over that. How do we isolate the DNA to do these? Well, you can take the DNA out of a cell, and then we would take some ethanol, add it to the solution, and the DNA actually just precipitates out, and it's kind of like cotton candy. It's spooled onto this glass rod. We use restriction enzymes to cut the DNA into fragments, and now we'll take that DNA and fragments and load it into what's called a well. And you can see in the bottom right hand corner there's a picture of a well, and really what it is a container full of clear jello called agarose, and the DNA goes into little holes in that gel, and if we apply a current to it, we'll see the DNA actually move up the plate because it's attracted to the positive pole. Longer strands, meaning the longer, more base pairs, move slowly because they're bigger, and the shorter strands end up moving further away. So here's that picture, an example of the wells, and you can see in those little vials there's little chunks or fragments of DNA being loaded into those wells, and you can see how the longer fragments stay at the top and then the shorter fragments go to the bottom. And so here's another depiction or graphic showing how those DNA fragments at the top are loaded into the gel. So if we took that gel and stood it up so you could see it, you could see that the DNA moves towards the positive end, and the shorter fragments are now at the bottom and the longer fragments are at the top. So here's a good picture. This is what an actual gel electrophoresis would look like. You can see all the many bandings. Usually the very first column is a baseline, so we're trying to compare it to the other strands. And if you look here, it looks like this one matches the last column. So if we were trying to, DNA fingerprint somebody or match this up with somebody, you'd say, well, the last DNA column definitely does match this baseline. So that transitions really nicely into DNA fingerprinting. And why would we do it? And usually you would do a DNA fingerprint to identify somebody for a crime or paternity, or maybe it's an inherited disorder. Identical twins are the only ones who would have identical prints because they come from the same sperm and egg. And a zygote formed after fertilization splits into two accidentally. So Here we can have two cell masses which are formed identically. And if I look at this DNA fingerprint, you can see that the P would be one of the parents, and then you could see, or not the parents, or maybe it's a suspect. So you can see P here would be the baseline, and I need to match it to suspect 1, suspect 2, suspect 3, or maybe child 1, child 2, or child 3. If I was to match this, what would you pick? Well, in this case, it looks like I'm going to pick suspect 2. So if you look at suspect 2, all of the bands are matching. Again, what are the applications for DNA fingerprinting? So paternity and maternity determination, making sure that the banding patterns match up, and you have half the mother and half the fathers. Maybe it's used for identification for criminals and forensics, or maybe just even personal identification that people would use their DNA for. And it looks like that is it. So the rest of the slides here are questions and extra examples on DNA fingerprinting. So go ahead and do those. Again, if you have any questions, you can remind your teacher or ask them in class.